So as I'm sure as almost all of you are aware and have experience with, when you're trying to secrete a protein in yeast, sometimes you're very, very lucky and secretion goes well, you get a lot of protein. And other times you're less lucky and you're in for a very long and laborious process of trial and error trying to optimize your protein secretion. Unfortunately, there are not really a lot of rules and models that can help you with this, so it's often really a process of trial and error.
Turn the volume.
We believe that this is due to the fact that most studies on the secretory system, so most fundamental studies on the secretory system, they only use a very small set of test proteins. And yeah, this data is of course very, very interesting for us, but it's oftentimes very difficult to generalize these studies to your protein of interest, because your protein might behave differently. And then secondly, if you would like to generate a very large data set of secretability, there are not really a lot of techniques that are able to do this. Most techniques, they lack like the breadth and complexity that is needed to construct these very big data sets. Okay, so to solve this problem, we generated a new technology which is called Sacrify. And Sacrify is an abbreviation for secretability screening of recombinant fragments in yeast. And Sacrify is basically a technique, a high-throughput technique that can generate secretability data for millions of proteins or protein fragments. And this slide summarizes how it works. So in the upper left corner, you can see our vector design. So in red, you have your library of interest. We are using a fragment library, but you can put any kinds of proteins in there. And then you can also see the vector context that we are using at the moment. We're working with the AUX1 promoter and the mating factor alpha as a secretion leader, but of course you can change this vector to your liking. Then we also have an N-terminal flag tag and a C-terminal V5 tag for detection purposes. And last but not least, we have a fusion to the SEG1 protein or part of the SEG1 protein. And this is very important because we will use surface display as a proxy for secretability. And it works like this. You have your library of proteins or protein fragments in our case. You transform them to yeast. And then if a protein can get secreted, it will travel throughout the secretory pathway. But because it is fused to the SEG1 protein, it will be retained onto the yeast cell wall. So you can see that in the lower right corner. So SEG1 will make sure that your protein is stuck to the cell wall. And this makes it very easy for us to sort out the cells that could secrete a protein via MAX or FAX. So you can see a graph of a FAX plot, and then because we have this N-terminal flag tag and C-terminal V5 tag, we can easily sort out the double positives. And these are cells that contain a fragment that was secretable. So then if we use next generation sequencing to determine what the DNA sequence was of this fragment, and if we compare this back to our original unsorted pool, we can get very big data sets of proteins which were enriched and so probably secretable, and proteins which were depleted in our data sets, so probably not secretable. Okay, so in our first study that we did with this new technique, we made use of a protein fragment library. We made this choice because we thought that when you're trying to secrete or express a protein, it often fails because of a very small patch or a very small sequence inside your protein. So we really wanted to go to the smallest relevant unit, which we thought was a folding unit, so a domain. So what we did is we used RNA, and we made cDNA, and then we made, using random fragmentation, we made a library that had a size distribution that corresponds to about the size of a protein domain. So on the graph on the left, you can see a size distribution for human protein domains as we got it from a database. And we were trying to emulate this a bit. So we made a library for using Saccharomyces and also a library for using Pichia. So those are the two other graphs. And yeah, you could see that in the Saccharomyces library, we still had a small bias towards smaller fragments. This was mostly due to PCR errors that we had. But we solved this issue when we made the library for Pichia pastoris. And then, of course, we had our library. Okay. And once we had our library, we did our Sacrify technique, and we got our first secretability data out of this. So we screened a lot of fragments. In total, we had about 100,000 fragments that we screened for Pichia pastoris. of which about 8,000 turned out to be secretable, and 110,000 turned out to be not secretable. Of course, we had finally this first very big data set of secretability, and we decided to try machine learning to find some features. So we used two techniques. We used both gradient boosting and convolutional neural networks. And then on the first, the upper graph on the left, this is kind of a plot that measures the accuracy of your model. And ideally, you would see this graph go up, straight up, and then straight to the right. And then you would have a perfect model. But in our case, it was already quite good. We could reach an area under the curve of about 0.78. Also, what's kind of interesting is that both techniques, even though they're working very differently, they were able to, they had graphs that were very similar, so they probably learned similar features, and both were very performant. Then in the graphs on the lower side, you can see a Pearson correlation plot. So here the models that we got from both techniques, we compared them, and you can see that they're correlating quite well. So once again, they probably learned the same features. And then lastly, we also tried to find out exactly what the features were that our model learned, and that we tried to plot on the colorful plot there on the right. So what we did is we plotted the amino acid sequence with the relative position inside the sequence. And if it's in blue, then it means that the amino acid was not favored for secretion. And if it's in red, it means it was favored for secretion. So now in our case, you could see that basically the position inside the sequence did not really matter all that much. Your amino acid was either favored or not favored. And also the features that we got from here were also not very surprising. For example, the small amino acids, such as serine and threonine, they were favored. And the very big, bulky, hydrophobic amino acids, they were not favored. So when we saw these results, we realized that we probably learned more like features of how you can escape the quality control in the secretory system, rather than really learn structural features. So we decided we had to take the next step in our library design, which is a real protein domain library with correct domain boundaries, so no longer using random fragmentation to get pieces that kind of have the same length as a domain, but we really wanted to use real domains. Okay, so then the first thing that we had to do was, of course, find out what a domain is. And luckily, other people have already spent a lot of time on this. There are a lot of domainome databases. And the first one that we thought was really interesting was the CAT database. So this is a protein database which classifies proteins into domains, but it uses PDB structures as input. So this means that it uses experimentally validated structures as input, and it's also manually curated at the end, so also a person takes a look at it. So we thought that this was a really good set to start with. It's probably very, very trustworthy. Of course, as you will see later, the CAT database does not contain a lot of protein domains. In our case, we're doing a proof of concept for the human protein domainome. It only contained about 5,000 human protein domains, so we wanted to enlarge our set a bit so that we had enough data to train deep learning models. So then we looked into more alpha-based prediction databases. And the first one we found was the CAT AlphaFold database. So as I said, the input here are AlphaFold prediction models. So it's also structures, but they're not experimentally validated. And then what they did here in this paper, they actually just ran the CAT algorithm on the AlphaFold models, and then used that to also generate domains. And then lastly, we also used the DPAM. This is also very similar in the way that it also uses AlphaFold prediction models as input. But it's not using the CAT algorithm, it's using the Eeckhaut algorithm. We also wanted to add this so we didn't have a bias towards the CAT algorithm. Okay. Yes, thank you. So yeah, this is an event diagram with our new library that we ended up with. So as you can see, there were only about 5,000 domains in the CAT database. And then the CAT AlphaFold and the DPAM database both were able to enlarge our database quite a lot, because in the end we ended up with about 40,000 protein domains for the human protein domainome. But luckily you can also see that there is quite a lot of overlap between all the different algorithms and databases. So this also was a kind of a good sign that not every algorithm or database was just basically doing its own thing. So then we started using this, and then after doing some more filtering, but this was mainly because a lot of protein domains, they're very similar, and then we only wanted to keep in one one protein domain that could sort of represent the others. So then after some additional filtering, we finally ended up with a library, an in silico library, of course, of about 28,000 human protein domains that we wanted to construct. Okay, so now we have our in silico library, but of course the next step is how are we going to generate this in the lab? So we did a deep dive into the literature, and we found out about a technique called dropsynth. So dropsynth is a technique from the Sri Kusuri and Kelen Pleza lab, and it's able to generate large libraries, DNA libraries, from low-cost oligopools. So this slide summarizes what it does. So basically you have to start from bioinformatic library design. So you have this pipeline, and you can feed all the things that you want to synthesize into it. It will chop up your protein, the DNA sequence of your protein, into smaller pieces, which are called oligos, which are single-stranded DNA sequences. And these can be synthesized by multiple companies into one big, large pool. So it's based on arrays, the way it is constructed. So then you end up with your one-pot oligo pool, which contains all the oligos you need for all of the DNA sequences that you want to make. Now, of course, how are you going to make your proteins out of this pool? So for that, you need your barcoded beads. So it works like this. So every oligo pertaining to one specific protein or protein domain that you want to construct has a unique barcode in there. And then one bead in your barcoded bead pool will also have that unique barcode on it. So in this way, you can target all the oligos that you need for your assembly to one specific bead, which means that they will be in very close proximity to one another. And so you actually just do a hybridization between your oligo pool and your beads. And then after you do this, you make these water and oil emulsions. So you get these water droplets in oil emulsions, and you try to do this in a way such that every water droplet only contains one bead. And then, of course, in your one droplet, you also only have the oligos needed for your one domain that you want to construct. Then you do a digestion reaction, so you can cleave off the oligos from your bead, so that the bead is no longer interfering in your reaction. And then finally, you can do your assembly. So what I did not mention yet is that during the bioinformatic process, you make sure that there are overlapping sequences. So every oligo overlaps with the next oligo that you need. So then basically you can just do a PCR reaction to fill in the gaps. You get your double-stranded DNA, then you break your emulsion, and hopefully you end up with the library that you wanted. Okay, so a few things that we changed to the protocol from Kelen Pleza is that we added some QC steps, some quality control steps. So first of all, we implemented capillary electrophoresis to follow the reactions to make sure that everything is working perfectly. And then finally, I also wanted to show something like just a normal agarose shell, of course, you can also use for this purpose. And you can see that the oligos, once they are assembled, you get this higher band, and you can very easily use gel extraction to cut it out. Thank you. Okay, and then the main drawback of the dropsynth technique as it was right now is that, of course, your oligos, they have a lot of errors in them. And when you then use all these oligos to construct larger fragments, the errors accumulate, and at the end you only end up with about 13% of your library that is actually a correct sequence. There are a lot of deletions or substitution errors in there, and of course you cannot really use these sequences for your purpose, but you also cannot easily get them out again. So you're always dragging along this very big weight in your library. And then luckily we found out that there's also such a thing as hi-fi oligos from Agilent. And when we use these oligos, actually the percentage of correct sequences in our library improved drastically from 13% to about 53%, which is of course very, very interesting because you're no longer dragging along this very big weight of unusable sequences inside your library. And then finally, so this is all still on the human protein domain. So finally we then looked into how many of these 27,000 sequences that we wanted to construct, how many we had actually constructed. And we had about 17,000 in our library. So this means that we had about a success rate of about 62%, which is quite nice. Of course, if this would be an issue, you could always try to improve this by constructing a second library. You then do the entire protocol again, and then by using different code and optimizations, different overlapping regions, you can kind of hopefully overcome the errors that happened here in this library, and then you can combine them. Okay, so here are my take-home messages. So my summary. So Sacrify is a platform technology for secretability screening in yeast. At the moment, it already generated the largest resource on eukaryotic secretability data. Okay. So we use Dropsend to generate a yeast surface display library of about 17,000 human protein domains. Our next steps are to use these libraries in our Sacrify assays. So we have a new data set on secretability, and hopefully we can further improve our deep learning models. And then finally, in parallel, we are also employing the same approach. So we also want to construct domainum libraries for other organisms, such as Arabidopsis, Drosophila, Saccharomyces, Plasmodium, and Trypanosoma. Okay, so this is the end of my presentation. I would like to take this time to acknowledge my lab, so the lab of Professor Nico Kallewaert, all of our alumni, and in particular Morgan Bone, who initiated the study and who generated the first data set on secretability, and Jasper Zualaert, who is our deep learning expert and helps a lot with all the bioinformatics aspect of this project. And also, thank you for listening.
Thank you for your nice talk. I just got a question. So how do you translate the oligo part to the protein part? That's not completely clear to me.
First you generate your library on a DNA level, and then you clone it into the surface display vector that I was talking about in the beginning, and you transform that to your yeast cells, and then they will generate the proteins, so they will try to secrete the proteins.
So you're going to secrete a whole bunch of proteins there, is that right?
Yes. So indeed, like in our first iteration of our technique, we screened millions of proteins this way. Okay. So you just transform this entire library, this vector library, to your cells. You try to aim that only one specific DNA sequence is inside one cell. And then you have this library of cells. Each one is expressing or trying to secrete one specific protein, and then you can screen it with MUX or FOX.
I see. Thank you so much.
Thank you.
Very nice talk. Maybe I missed it. What's the correlation between something being secretable in Cerevisiae versus Pichia, or Pichia versus Cerevisiae? Is there kind of like a...
I did not touch upon that, that's true, but actually we saw the same features popping up when we looked at the primary amino acid sequence. It was very, very similar. But of course, our first library was generated with a fragment, so with protein fragments. So we think most of these fragments were unfolded or not in a correct fold. So we were probably more indeed screening for features that could be able to evade QC in the secretory system rather than folded proteins that can get
So were
secreted.
they actually all open reading frames that were expressing your tag?
Yes.
Okay, so 90% of them didn't work in that case.
Yes, exactly.